Okay, so I remember now where I got to. So the Mars colonization enthusiasts, they say that they are likely to try and argue with the lawmakers. So lawmakers are going to be are going to say are going to look at all sides of the argument, and so you're going to have the astrobiologists and the general public, if they're concerned about life being returned from other planets, then during this long legal process, which would last at least a decade, then they're going to be saying, we've got to show that it's safe for Earth life. Now, the main argument that all the arguments of the space colonization enthusiasts, or all Robert Zubrin's arguments about how saying that it's like trying to, he has this argument about smuggling Canada geese from the United States to Canada when they can just fly. But those sort of analogies aren't going to work when you're talking to lawmakers who are worried about whether microbes from Mars could be harmful to Earth, because they're not Canada geese. And it's obvious that you can't just use an analogy like that to convince them that the Mars life has got here already. And there's the same problems coming back from Mars to Earth. We don't know. Mars life that could be harmful to us might still not be able to escape from Mars. The most likely place for the samples that we're currently getting from Mars back to Earth, they are from impacts into the high southern uplands, because that's when there's much less atmosphere between them and space, and so it's easy to knock stuff into space. It's very dry, it's very cold, and it's not the most likely place, like right up Olympus Mons or something like that. But that entire upland region, it's not the best place to find Martian life, and the rocks that are getting here from Mars are from that region, and they've spent hundreds of thousands of years, the ones that are arriving just now, and most of them millions of years, of the ones arriving just now in space, thoroughly sterilized. All the Mars meteorites that are arriving on Earth right now are definitely thoroughly sterilized. There's no life in them, because they're just not big enough to withstand the ionizing radiation from cosmic radiation and from the sun, kind of just sterilizing it, and solar storms sterilizing it during the journey from Mars to Earth. And it's not at all clear that any Mars life would actually get here, because we don't know what its capabilities are. We don't know if it can withstand the shock of being thrown up into space. We don't know. It's quite easy. And how would it get into the meteorite, into the material? Because most of the habitats there are very fragile. They're like dust and salts and ice, and like salt on top of ice and dust and clay and stuff. How much of that would actually get into space after an impact of a large meteorite hitting Mars and throwing material into space with enough velocity to get to Earth? And for different reasons, it's not certain at all that any Mars life has got from Mars to Earth, because we don't know what its capabilities are. And it could be perfectly fine, but just not be able to resist whatever it takes. And for instance, we have a lot of photosynthetic life on Earth that is simply not able to withstand the shock of ejection from Mars. It's just too much. And that's a paper by Charles Cockle. He found in the forward direction from Earth to Mars it's almost pretty much impossible. From Mars to Earth, then it's also very difficult for photosynthetic life. So if only photosynthetic life, most of it just can't get from Mars to Earth, then what about other forms of life on Mars? I mean, there's no reason why they should be able to withstand shock. It's not a thing that it naturally has to face. It's more or less an accident of evolution if it's able to get from Mars to Earth. And then it's got to withstand going through the Earth's atmosphere and the heating up of the skin, if there's anything on the skin of the meteorite. And once it gets here, of course, then it falls in the sea or something, then that's the easy part. It's the other way around from in the other. The shock is the same, problems both ways. So anyway, these arguments about Canada geese would not convince the lawmakers that all Mars life that could harm Earth has already got here on meteorites. It's not even certain that some of it has. and certainly not all of it. So they're not going to be convinced by those arguments. And so, or even though it's very convincing, deeply convincing, when Robert Zubin gives these arguments with planetary protection officers, and he makes these points, and the planetary protection officers make their points, planetary protection spokespeople, not officers, I haven't actually seen this office debate directly with him, but you get some planetary people, experts, try to debate with Robert Zubin, and he's a very, very strong, he can argue the hind leg of a donkey. But then, I'm rather like that as well. I come from a family of very argumentative, and we love arguing about all sorts of things. So you develop these kind of skills in arguing, but it doesn't necessarily mean that what you're saying is right. So you have to be careful about what you're saying is correct, as well as being very effective in arguments. So you have valid and invalid, and you have persuasive argument based on pathos, and logos of trying to use authority, of ethos, using authority, and then logos of the kind of reasoning argument. And I try to focus mainly on the reasoning, but with some vivid images and things to give a little touch of trying to help the argument along. And there's nothing wrong with that. So so anyway, in a law court, then just vivid images like talking about Canada geese is not going to persuade them. And these people are not going to be particularly disposed to saying that we've got to let the Mars sample be returned to Earth because we have to colonise Mars, which is what these colonisation enthusiasts are likely to say. And their reason for saying we have to colonise Mars, they tend to say we have to go to Mars in order to have a second home for humanity, because they sort of seem to think that Earth could become uninhabitable, and yet the Mars could be habitable. It doesn't really make much sense when you think about it carefully. And we're doing very well on Earth. We're falling. It's remarkable how we're pulling together. We've saved the whales. We've had the Green Revolution, which saved billions of people's salvation. We've saved the ozone layer. We've eradicated smallpox. We're doing huge numbers of things all together. We've managed to continue to manage to feed everyone with a food surplus, a 30% grain food surplus every year, retained for the next year. Every year we retain 30% of grains for the next year, even with a rapidly increasing population. As the population increases, our food is also increasing just as fast. And the main population increase in the future is going to be in sub-Saharan Africa, and they have a potential for a tenfold increase in the productivity because they missed out on the Green Revolution. So even with a fourfold increase of the population, they have plenty of leeway to feed everyone if we can have the equivalent of a Green Revolution in sub-Saharan Africa. And it's well possible that it levels off before then, if the key to that is education and good infant care, and reduce infant mortality, and good health care, and education of the women is very important. If we have all of that, then it's likely that because our population is levelling off due to prosperity, when people are wealthy, then they invest more in fewer children, and many of them, with a well-supported society around them that supports them, are perfectly happy not actually having children. They don't feel you have to have children to support you in your later life. And so that's the situation that we are, and we are actually, I've helped these people who are very scared of various doomsday scenarios, and so I've looked very deeply into all the suggestions of ways that civilization and human beings could be harmed, and I just don't find any possibility of human extinction, and I don't find any possibility even of collapse of civilization. We're far more robust. I think the only returning life from another planet, as I was in the middle of saying in the last one, that if way back in the Apollo times, if there'd been harmful microbes on the moon, then I think we did have a risk of extinction then. And I think that some other species may well at that stage have become extinct. But we've managed to move far forward in our awareness since then, and I don't think we have any risk from that now. because I think, as I say, we're going to look very carefully at stuff like this. We're not going to just return something willy-nilly from Earth, no matter how much the space colonization enthusiasts say. And I was saying we're actually far safer, even if it in a very remote case, because we could identify it and work with it in ways that weren't even remotely possible at the time of Apollo. So we have, I think that would be perhaps not nuclear war. I don't see nuclear war. I see returning samples from the moon as being the kind of touch point in the past, with my background of now understanding a little bit about planetary protection, where we were maybe a bit lucky. Anyway, and then in the future, then a very large asteroid impact could be pretty harmful to us, but we know all the big ones, all the ones above 10 kilometers. We know nearly all the ones above 1 kilometer. Long period comets are very, very rare. We get plenty going through the solar system, but even with their huge tails and so visible in the daytime comet, like Hale-Bopp, then these very bright comets, they're still further away than the sun. They're vast distance away, and they're just so obvious because they're so big in the sky, because their very, very flimsy tail spreads across a large area and reflects so much sunlight. And the nearest any comet, long period comet, rather than the short period comet has come, is Lexell comet in 1770. So, and that's 6 times the distance of the moon, and I really don't think we need to worry about long period comets. But if you are thinking about this, we're going into space because we're worried about what the comet might do to Earth, then you don't set up a colony in space and say, okay, we'll just let this comet do what it's like to Earth, and we'll live somewhere else instead. Because what you'd do is you'd make sure you have lots and lots of telescopes to detect these comets in years in advance, and then you have an infrared laser array. It's one way to deflect them so they can't hit Earth in that very, 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 very remote possibility where you're probably waiting at the current rate of comets going past Earth. It must, if we were hit by comets in the past, it was surely at a time when there were more than there are now. Perhaps as a result of a star dispersed going through the Oort cloud or something. It doesn't seem likely that we were hit in the past by comets if it was current flux, not the long period ones. The short period ones, yes, maybe, but not those long ones. So if the dinosaurs were hit by one, it was probably because of some perturbation of the Oort cloud before it. But there's the possibility. There's Swift-Tuttle in 4479, I think it is, and there's a 1,000,000 to 1 chance, but Swift-Tuttle could hit Earth. But obviously, in those couple of thousand centuries, couple of thousand years, then what we would do in that very, very remote chance, 1,000,000 to 1 chances, means we don't expect it to happen. Definitely extremely unlikely, to the point that you just sort of almost forget. 1,000,000 to 1 is, despite the movies, that's a situation where you just say forget about it. If we don't expect that to happen. But in that very, 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 very remote chance, then we've got 2,000 years to figure out what to do about Swift-Tuttle. And there are these methods which can deflect a comet in just 2 years. If we can build a laser array that works for just a few minutes every day. So I think that if we're worried about preserving humanity, we've got to keep Earth in a good shape. And if you're worried about cosmic things like comets and asteroids, then have telescopes and then work on ways to deflect them, and that's going to cost far, far less than trying to start up a colony on Mars. So I don't think this is a good argument, and I can't see them persuading legislators that we've got to drop protection from Earth in order to set up a second colony on Mars. It's very convincing to other space colonization enthusiasts, but not much to anyone else. I think they live in a rather close little world where they're talking to each other, and they all have this great belief in that colonization is the future. Now, I'm very keen on space colonization, so I think we should go to the moon. And we should have a lot of comparison of the two, of Mars and the moon. I find that the moon is greatly favourable. I didn't expect that until I did that work I've got this, I've read, reading the literature, not my own research obviously, but reading the literature on the topic and then writing it up for my case for Moon first. And as Chris Hadfield said, it's our generation is the Moon generation. And then the babies of today, just being born today, they may be the Mars generation. But not just Mars. I think by then we'd be going off to places like Callisto in Europe, and not humans to Europe, but Callisto is the best place for humans, and then Titan, and we'd be using materials in the asteroid belt, and maybe Venus clouds, certainly have some aerostats there. And so I think we're rather too laser focused on Mars at the moment. And we can even do astrobiology on the Moon. Humans can go to the Moon, and the lunar poles of the Moon have received samples from around the solar system, and certainly from Mars. There should be early Mars meteorites at the lunar poles, and if there's ice there, then well below the surface there should be intact organics from Mars, and it may well have preserved even intact DNA. And we should have bits of dinosaurs, or maybe not dinosaurs, maybe more like ammonites, little bits of whatever the Chicxulub impact, it went into a shallow sea, and sure there were lots of ammonites there, and little bits of them should be at the lunar poles, and little bits of microbes certainly, and diatoms and things, and that again might have intact DNA in it. Not quite viable because it would have had some ionizing radiation, but if it was well dug deep below the surface at liquid nitrogen temperatures, it's so cold there in the lunar poles because it's got an axis that is pointing almost vertically to the sun. So there's some craters that are so cold that they would certainly preserve organics for billions of years. And we might even have life from Venus, according to some people who think it might have had a thin atmosphere early on. And these are not my ideas, this is from papers that have studied the possibility of finding meteorites, and also the possibility of finding intact diatom shells, that you find that the impact on the Earth would preserve the shell intact, a very small, almost intact. I think they even can get intact the smallest diatoms. And so you could find the shells there. And so we can do astrobiology on the Moon, and there's quite a lot of it, actually a significant amount of that material that we should eventually find as we sift through the ice, if there is thick ice there. So and the Moon, and if you want to have a backup for the seeds on, like for instance, the lunar poles at liquid nitrogen, passive cooling to liquid nitrogen temperatures, that's absolutely perfect for a seed bank. Seeds at those temperatures can last for centuries to thousands of years. And we would have to drill a hole well below the surface. We could start a seed bank with an early lunar mission to the Moon. Just take a pack of some interesting seeds from Earth and drill a little hole and drill down a couple of meters and leave the seed at the bottom and then fill it up. And then come back 1,000 years from now and you should find viable seeds there without having to do anything to keep it at those temperatures. So the Moon is a great place for a backup. So if you're going for a backup of things that are on Earth, so we already have these backups on Earth, like the Norwegian Svalbard seed vault. Well, if with easy heavy lift back and forth to the Moon, then we can have a backup like that at the lunar poles. And then that'd be very secure no matter what happens on Earth. Then these seeds are just there, we can forget about them. We don't need to do anything to keep them cool. And 1,000 years later, you can go back to the Moon and get these seeds that, if for some reason something has happened to seed banks on Earth, it's literally send those seeds to the Moon and forget about them, and just have a list of what you left there, and it's still there 1,000 years later. So I think the Moon is the place if you want a backup for in case of things happening on Earth where you might lose some important genetic material, as long as it's seeds and things like that. And if you want to have human beings there in a small colony of say 100 and looking after some of the rare species that's there as well in kind of greenhouses, and some of the plants like mangoes that can't be kept in seed vaults. So I think that is a far more sensible thing than going to Mars if your object is to looking at Earth and human biodiversity and involving it in the whole thing of making sure that you have backups, which we already have on Earth, and we want to have an extra one on the Moon, and of our knowledge and the libraries and things. So I think there's some point in that, but I can't see that argument as working as a reason for returning samples from Mars. And there are plenty of reasons to have humans in space, but that's not a good one. And so we come to all these, and there's just no way. They're just in dreaming, really, if they think that they can somehow persuade legislators to bypass legislation in order to let them return something from Mars. That's not going to happen, not nowadays. There are all these things you have to do, and I say if you've got spacecraft going to Mars and coming back again, then it's going to be returning dust from Mars. And if we can actually have enough awareness that is really a significant risk, it's not just people doing paperwork. If there is Martian biology and it's different from Earth life, it could be a serious risk for the astronauts as well if they were to land on the Mars surface. Now, it could be totally safe. Maybe it is. Maybe there's nothing in all this. But we don't know that, and we won't know that until we've done in situ searches on life on Mars. We can't tell that by returning samples, because that they just can't do anything even remotely resembling a kind of thorough study just by returning a sample at $10 million a gram from time to time from Mars. That's not going to tell you much. And so I think that if only the space advocates can understand this. And of course, tell me if there's anything. I've talked about this frequently with other people, and so the reason I have a lot of confidence in this, in what I'm saying, is not because I have a lot of confidence in my own reasoning, but because I've argued back and forth and talked to many people, and nobody has found any answers to this that I've come across. So think it over and see if am I right, or is there something I'm missing, and if there is, then do let me know. And I'm not the only one, as I say. There are these astrobiologists who have written these Mars sample return reports, and people who've written books and things and papers, but I think that my main take on this, which is why I want to try and get something published, is this the potential severe effects of biosphere collision. I'm not sure that it has that the severity of this is quite, that the astrobiologists are quite, they seem to be almost pulling their punches in the planetary protection discussions. There's it's just sort of they don't go all the way to saying how actually severe it could be. So cloud just goes in the forward direction, but nobody's using it in the backward direction. And I think we need to spread awareness, at least discuss this, and see whether there are these potential severe outcomes. And we need to know this. And the best way, I think, is to search for life in situ on Mars and understand it well, and then make any decisions about what we do next, depending on what we find there. And personally, I find it hard to think of anything we could find on Mars that wouldn't suggest that we at least need to take some caution. Because even if there's no life there, if there's Earth life there, and it was sent there on Chicxulub with that asteroid and remote possibility, we want to know what has happened to it in 66 million years. How is it adapted on the Martian surface? How is it different from Earth life? Was there any previous Martian life there? If there was previous Martian life, is it in a shadow biosphere? We don't want to mess all that up with masses and masses of Earth microbes before we can even know what was there. Even if it's very closely related to Earth life, but 66 million years on Mars, it could be very interesting to know what's happened in the time in between. And then, and you could tell us loads. And then the other possibility, suppose that that what we've got on Mars, suppose Charles Cockle is right. He doesn't say that it is like this, it's just an hypothesis. Suppose there are uninhabited habitats on Mars, places where Earth life could flourish, and where Mars life could flourish, but there isn't any. And that's quite plausible because the various possibilities that Mars life could have gone extinct even if there was early life, Mars life, and maybe it hasn't started up again yet, for instance, or maybe there never was Mars life there. Look at his paper, Trajectories of Martian Habitability, and he looks at many different possibilities. So, but if Mars is uninhabited, but it has habitats, that is, in my view, and I'm just presenting something to think about, that is amongst the most vulnerable thing you could have on Mars. Because when you think about uninhabited habitat, that means uninhabited by life. But what about prebiotic processes? Like the idea of an RNA world. So the various ways that RNA could maybe form in Mars conditions. Well, if there never was any life, what is the RNA there? Are the fragments of RNA there that have formed in a prebiotic way without any life there? That would be extremely interesting, but as soon as you introduce Earth life, you've lost any possibility of finding it, certainly if finding it easily, certainly in present day habitats. And suppose that you get these, you could have these RNA world cells that are very, very vulnerable, as I was saying. You could have something even earlier than that, autopoietic cells. And these are cells that are almost alive, but they're not really. They would even have RNA in it, but if RNA doesn't replicate exactly, it's functioning more or less like modern RNA, but it's not actually doing, it hasn't yet managed to achieve exact replication. And maybe it's been like that for 4 billion years. And you've had stuff that is almost life, and you have these little cell-like creatures, things that are almost replicating, but they wouldn't count as life because they're not exactly replicating. And yet they're able to do something, and they work, and they're functioning, and they're able to do what they do because of previous ones. They may even have hardy spore-like things that can spread around in the dust and then land somewhere, and then they start modifying their surroundings, but they don't reproduce exactly. Is that life? And if there's none of that, if it is very boring chemistry, and there's no complexity there as well at all, then that also tells us something. That tells us that it's very, very hard for life, or even prebiotic life, to arise. So, I mean, it might be that if we find it's just very boring chemistry, it might be that we decide very quickly that there's nothing there really to protect. But even then, we would want to know that first, rather than introduce Earth microbes and then try and speculate about whether, before the Earth microbes got there, what it was like before. So I think we should really consider protecting Mars, at least for some time, in any situation, as long as there are potential habitats for Mars life there, Earth life there, and there seems excellent potential that there are possible habitats for Earth life there. Many of them, I say, more potential habitats than there have been years since 2008. Dozen, maybe more than that, depends how you count them. For instance, you could count every single kind of cave that could occur on Mars as a potential habitat, including the sulfur caves, for instance. By the way, like for instance, you could easily have acid-tolerant life on Mars. So the idea that the Mars life couldn't tolerate a human stomach doesn't really make too much sense. You would easily have acid-loving life on Mars that is grown in these chemical acid environments. And then there's the many kinds of caves that could occur on Mars, actually more variety than there are on Earth. And another thing I'd like to talk about just briefly, because it's kind of interest. I mean, I think I've made my main points now in both the forward and the backward direction. Just a kind of interesting, just talk a little bit about the Viking and what it might tell about what we might find there. So the Viking lander, then with its labelled release, then, and I don't know what I'm going to do in terms of writing this up as a paper. I'd be interested in any thoughts you have about writing this up as a blog post. I mean, I don't really know. It's a matter of how you could select from this something interesting and not too long that you could present that could maybe get a bit of attention, because I would like this to be discussed rather more than it is, and try to find something a little bit eye-catching to try and get people just thinking. But my posts tend to be a little bit long, and is there something short that I could do that's not so long as my kind of articles that would get people thinking? So I haven't written this article. It's just a draft. I've been thinking about it for a week or two. I'm still not really sure what to do. But anyway, so the this is the Viking labelled release. So Viking, that's the 1970s. It was confused by the perchlorates, which are very reactive, and all the experiments on Viking, they came up confused. And the little ovens, which also you have on Curiosity, then if you heat up perchlorates mixed with the small amount of organics you could have if there's a small amount of life, then the perchlorates destroy the organics as it heats up. And just about all our ways of analysing organics on Mars to date have been through heating little ovens, which makes it almost impossible to discover tiny little bits of organics. And that was probably what happened why the Mars organic detection didn't work, because it was heating up perchlorates along with the organics. You only discover a very strong organic signal if you're doing that. I'm told by the people who analyse this. And then the Viking, it was quite strange, some of the things they did, but there's some things that were the labelled release. What this did is they fed microbes with a food that was radioactive, slightly radioactive carbon in the food. And then this food, if there was life there, the life would give off the carbon dioxide. And these black dots showed the carbon dioxide being given off in a kind of cycle. And this is very reminiscent of Earth life. It's a circadian rhythm, day-night cycle. And here's the temperature changing, because they didn't keep it entirely constant. It didn't vary as much as the Mars surface, but it did vary like that. And so as the temperature varied, so did the carbon dioxide given off. But if you look at this with expert eyes, and you look at the peak of the carbon dioxide, and so an expert in circadian rhythms looked at that, and he said that all the peaks are being delayed too much. You see there, it's gone up, and it's coming down like that, and they're delayed by 2 hours. Apparently, with normal chemistry, then 20 minutes could be explained, but 2 hours is very hard to explain with chemistry. So either you have something very complex chemistry going on there, or you have life. And so there's some. It's now no longer just Gilbert Levin now. There's some other astrobiologists and scientists coming around to the view that it's just possible that Viking did discover life there in the 1970s. And then if you think about it, Viking is the only one to have dug into the surface, dug down. And if you think about this, just as my own personal speculation, I haven't seen anyone suggest this, but this, if you go back to the idea of this biofilm that could exist in a region of 5 to 15 centimeters below the Martian surface, where you get these salty brines, and in the top 5 centimeters it reaches up to levels where Earth metabolism could survive in daytime, except it gets too salty. But if it was a Martian biofilm, maybe even an Earth biofilm, it could preserve the liquid from the night through to the day. Then if you take that, then then Viking could have sampled that, because it dug into the sand, and it could have taken up a bit of this Martian biofilm into its container. It dug deep enough. And it's the only lander since then that we've sent to Mars that has been able to dig that deep. Well, a Phoenix did a bit of digging, but but it was in a different part of Mars, and it didn't have any life detection. None of our rovers since then have life detection, and so the Viking landers, they may have discovered life. So if we go there, how are we going to find out? I think what we should do, I think that 2 missions, should be top priority for Mars. One we could do right away. We could do even, they haven't sent the successor to Curiosity to Mars yet. if they were, and I can't imagine them actually doing this, but if you imagine they had said we've got to have a rush job and put some life detection instruments on there, they've got two big things there that could be replaced by life detection. If they would decide we're not going to be bothered with a sample return, if somehow someone could be convinced by everything that the astrobiologists have been saying and I've been kind of talking about here, that actually a sample return is not going to work, and postpone it for another year or something, another couple of years, and actually put life detection on it, then that's plenty of enough mass to have loads of life detection instruments. They've also got MOXIE, which is just designed to generate oxygen, and it's absolutely not needed. It's not used for any of the other experiments. It's just going to be travelling around on the surface of Mars, generating oxygen and venting it into the atmosphere in order to find out whether we can create oxygen in Mars surface conditions. If you think that perhaps sending humans to Mars is not top priority and it's not going to be the very first thing you do, and maybe won't be doing that for at least 20 years, then it's somewhat less of a priority to be able to know if we can use chemistry to make oxygen in the Mars environment, which is what they are going to be testing with this. And it doesn't need to move. It could be stationary on any stationary lander to Mars. I don't really see that it's such an urgent thing that we have to do it now, rather than say 2 years from now, whatever. And that again is enough for a whole load of 4 or 5 life detection experiments could replace the mass of that. It's quite a heavy thing. So if we were to send, let's suppose, just kind of fantasy, that we're going to send life detection to Mars, but maybe, I very much doubt NASA will do it. And ESA, they're not sending life detection quite yet, but they are both sending Raman spectroscopy, which will at least not have those little ovens. It's not as good at detecting organics as specialized instruments designed for it. For instance, if the organics is mixed in clay, which is a very likely substrate, then it's very easy for this to get mixed up and not really be able to detect the organics very well because of confusing signals from the clay. So it's not the kind of if you're optimizing for detecting life organics, it's better than the ovens. It complements them, but it's not really necessarily what you would choose to send to Mars if your focus was on life detection. Not just that. You probably would have that, but it wouldn't be the only thing you'd send. And anyway, maybe the next mission after this, then maybe they detect something there that gives a signal that makes you think if ExoMars will be able to drill. So maybe it finds something below the surface that's interesting, and or maybe they do find some interesting organics that they seem like they could possibly be life. Then we send life detection, hopefully, rather than return a sample, which I say is not going to work. But if they find something there that looks like life, not just like a habitat, then sample return is a sensible thing to do. But if not, then it doesn't really make much sense. So anyway, if we're sending up there, with some future, we're actually going to look in situ for try and duplicate the Viking mission, and it's either them, maybe India does it, or China does it, or Russia does it, and or maybe private space. There's some keen billionaire who wants to find out and uses private space to look for it. Then we can do the same experiment and see if we get the delayed circadian rhythms. Another thing that Viking did, that when the sample was kept in darkness for a number of months, then you didn't get this signal anymore. And that's very difficult to understand in terms of chemistry. Why? If you took a sample of the Martian dust and kept it dark for several months, why would that so alter the chemistry that it no longer does this thing of evolving the carbon dioxide? Another thing they found is the carbon dioxide amounts were reduced as the temperatures went up. It's the sort of thing that would happen with life that couldn't tolerate high temperatures. But it's hard to understand that chemically, why you could have less carbon dioxide evolved at higher temperatures. So there are quite a few mysteries with the Viking landing experiments that really they need to be explored. What is it? Is it some very complicated chemistry, or is it life? and we need to find out. And so I think we should duplicate the Viking lander experiment with more modern techniques, modern instruments, to find out a lot more about what's going on. And one of the things we should do is to feed it with a chiral solution. So that instead of, what they did is they just, in order to have the food, they thought that it would be absolutely unambiguous, and that the others the other experiments would take organics, and then Viking would just find the CO2 evolved, and everybody would just say, oh, we found life there. So they weren't kind of designing against, making it bulletproof against things like this. They designed an experiment that would have worked on Earth. It worked in the Atacama Desert, it would work anywhere on Earth, but it didn't work on Mars. It's not too surprising for the first experiment you sent to Mars. But with hindsight, what they should have done is to have the amino acids in both the left and the right-hand side separate, instead of having them both at once, so that any conceivable extraterrestrial biology will be able to eat it if it likes organics and if it likes amino acids. They would have some of the left and some of the right, and they do experiment with the left, experiment with the right, they can do experiment with both, and then see what happens and compare how much carbon dioxide is evolved. Now, that is based on the idea that modern Earth life is that life would either always prefer the left or always prefer the right. But it could be that Mars life is achiral. Now, not many people actually think about this, but there's an enzyme called Joyce's enzyme, and this joins together the opposite chirality. So if it's left-handed enzyme, then it will join together right-handed. If it's right-handed, it will join together left-handed. And so this means that it can, it's a ribozyme, I think it is actually. And it can replicate itself. So it can replicate its mirror image, and its mirror image can replicate itself. There's a paper about it, and I can link to it. I haven't actually got that in my article here. I'm going to add a section about that. I forgot to add it, but so I've got in my Touch Mars ? Book. So you could have achiral life there that is going to be able to eat both types of food, and you could also have like the very early autopoietic cells, and they might be able to handle both kinds of food. So if you talk about, especially talking about early life, and I think of various other ideas, then it is possible that it turns out that it can eat both sides. So you need to have a whole spectrum of different experiments, and but that would be very interesting as well. So you do the two chiralities and you find that it is indifferent of them all, but in other ways it's still got these delayed circadian rhythms, and you're still wondering, is it chemistry or is it life? And then you can try, there's holographic microscopes you can use, where you make a hologram based on what you see on Mars, and that has the advantage that you can focus it back on Earth. You can recover a focused image from a hologram. That's called holographic microscopy, and this lets you look at, for instance, things swimming around in solution, and you'll be able to focus at different depths in the solution, to have one frame of holographic image, and you can focus and you can get things in perfect focus. And so some people who experiment in holographic microscopy have suggested this application. And there's lots of and it would be there's lots of these. I've measured some other way out things here. There's lots of ways lots of things. There's the astrobiome nibbler, which can recognize even just one amino acid in solution out of a sample, and there's polyclonal antibodies. There's the what you call them, the microbial cells, where you look for redox reactions, and the oxidizing and the reducing that you get in life, and what do they call them, microbial. It's got an electrical current flowing through it, and so the loads and loads, the astrobiologists have been so creative. There have been so many astrobiological instruments that have been designed to send to Mars. And again, I'm sure there's been more, certainly been more astrobiological instruments designed for Mars than there have been years since 2008, though many of them designed well before then. Like the NASA URI instruments, which sadly they pulled out of the collaboration with Europe, which was going to make it possible to send it to Mars, and so it's rather sad. And so, but there's a vast creativity and number of instruments we could send, and nowadays they're very low mass. You get things that are just the size of almost the size. You get a lab on a chip, it's called. We put tiny microfluids, sending little bits of fluid through tiny little channels on a tiny chip, and some of them are a bit bigger. You have you can have a gene sequencer sent to Mars that you could hold in two hands. There's a complete end-to-end, doing everything, collecting the sample, processing it, sequencing it, and then sort of emailing, as it were, radioing the sequence back to Earth. It actually does the entire process, working out the sequence, because sending the data back would be a vast amount to send back. So it's amazing the ingenuity of what they can do nowadays. And just a decade or so ago, then that would have been filled an entire laboratory. And now it's a little thing you can hold in two hands. So that's part of the reason that astrobiologists are so keen on sending instruments there, because they know that we can now send remarkable instruments to Mars that we could never have done before. Remember, you send your very best, the highest engineered instruments that you've got to Mars. And for instance, one of the ones, the polyclonal antibodies one, that one has found life in the Atacama Desert that hadn't been discovered before, because it's very good at discovering very, very faint signals of life, and it drilled 2 meters below the surface and it found life there. So you're talking about cutting edge equipment for finding life, and we could send to Mars and search in situ. So I think that's what we should do, and it's really, really exciting if we do this. Even if we, but you've got to realise it could be a null result. There's maybe no life there. If there's no life there, then that is tremendously interesting as well. Now that's a little bit difficult to sell to the public, but this is the way you've got to do it in science. You have to recognise that a null result is exciting. If there's no life there, if there's no sequenced life, then you know that if there's any life, it's not any DNA in it. Maybe got some other kind of way of replicating. And if there's no life at all there, then you know you've got a planet which hasn't even evolved life yet, or it's gone extinct. And then you can go and look and see what a habitat is like if it hasn't got life. Now, if you don't send life detection instruments there, you've got no way of making this discovery that there's no life there. And you've got to do that before you send humans there. I think if it's got uninhabited habitats, you want to know what they're like. So anyway, I think that's enough on this, and I've probably said enough. If any of you have any thoughts about, of course, if you have said anything wrong in any of this, please correct any errors. And if there's got any other thoughts about it, do say. And if you have any thoughts about how I could do a blog post about all this, because it's a bit of a puzzle. It's just too much, and I'm rather at a loss at the moment as to where I'm going to take this.